Hello, welcome to Biology Lessons with Dr. Lin. Today our topic is CRISPR-Cas9. CRISPR-Cas9 is one of the most important scientific discoveries in history. With CRISPR-Cas9, we can potentially make any changes we want to the human genome easily. We can potentially cure genetic diseases, fight cancer with much greater success, make humans live much longer, and engineer superior organisms, including humans. It all started with some bacteria that have been frequently attacked by viruses. These bacteria evolved a defense mechanism, CRISPR-Cas9, to protect themselves from the viruses. When a virus injects its DNA into the bacteria, a segment of the viral DNA is removed and inserted into the bacteria's own genome in a region called CRISPR. This region of the bacterial DNA is converted into a piece of RNA called the guide RNA, which is combined with the Cas9 protein. Cas9 is a DNA scissor and can cut DNA. The guide RNA leads the Cas9 protein to another copy of the viral DNA inside the bacteria. The guide RNA can find the viral DNA because its sequence matches the sequence of a portion of the viral DNA. Once Cas9 reaches the viral DNA in the bacteria, it acts as a DNA scissor and cuts the viral DNA. The viral DNA is now damaged and can no longer function. This is how some bacteria neutralize the threat from the viruses using CRISPR-Cas9. After scientists discovered this defense mechanism against viruses in bacteria, they realized that they can use the same mechanism to make any changes to the DNA that they wanted. How? Well, they can genetically engineer the guide RNA to match any sequence of the DNA genome of an organism, such as humans. The guide RNA takes Cas9, the DNA scissor, to that DNA sequence, and Cas9 cuts out a segment of the DNA at that sequence. Normally, a cell would repair such a break in the DNA by inserting the broken piece of DNA back into the gap. If, however, the cell is supplied with a piece of genetically engineered DNA, the cell may insert this engineered DNA into the gap instead. The genetically engineered piece of DNA is created in the lab and can contain any DNA sequence that we want to insert at that specific location in the genome. Thus, we are potentially able to make any changes we want to the human DNA anywhere in the human genome using CRISPR-Cas9. Thank you for watching Biology Lessons with Dr. Lin. Please like, comment, and subscribe. Hit that subscribe button and get notified when the next video is posted.